in the, I forgot, the institute.
Yeah. For history and philosophy of science and technology.
Perfect, that's in Paris. And we are very excited to have Francesca here because she thinks she's a philosopher that thinks about evolutionary biology with a strong emphasis on chance and inheritance and epistasis, and we're very much looking forward to see what you guys are thinking about all these concepts and topics. So thank you very much for coming.
Thank you very much, Luisa. Thank you for inviting me to all the organizers. Thank you for being here. So in this talk, I will try to convey some philosophical issues raised by the study of stochasticity, what you do, the study of stochasticity in biology. And mostly it will be the occasion for me to ask you to raise questions, yeah, because I would like to discuss them with you, and also to, at the end, to suggest a sort of philosophical proposal I have been elaborating with my former PhD student, who in fact is going to give a talk just after me. So, but first of all, before going into the talk, I would like to say some words about philosophy and philosophy of biology in particular. So what is it? What's the aim of philosophy? I like to answer by using a famous quote by the American philosopher Wilfrid Sellars, saying that, I quote, "The aim of philosophy, abstractly formulated, is to understand how things, in the broadest possible sense of the term, hang together in the broadest possible sense of the term." So he's saying, in short, that philosophy looks for the big picture. And I want to say another way to say that in more recent terms by the philosopher of biology, Peter Gottfried-Smith, is that philosophy looks for, I quote, an overall picture of what the world is like and how we fit into it in an especially broad way. So philosophers look for this big picture, for what is the furniture of the world and how things hang together. And Peter Gottfried-Smith also, I like this distinction he makes. Now we are talking about philosophy of biology. There are two different ways of doing philosophy of biology, of conceiving of philosophy of biology, and you can do the same both at the same time. So philosophy of biology can be interested in the living world, and so in this case, science is an instrument, one of the instruments to study, understand the living world, and so it's philosophy of biology as philosophy of nature. And also, philosophy of biology can be interested in the human investigation of the living world, which is science, what you do. And so science is a subject matter here. And philosophy of biology is a meta-discourse on science. What is interesting, when I read this paper by Conrad Waddington, so it's a paper presenting some of the points discussed at two meetings in 66 and 67 at the instigation of the International Union of Biological Sciences. So in this paper, Waddington, just at the beginning, is trying to define what is theoretical biology. And he says, I quote, "Theoretical biology is an attempt to discover and formulate general concepts and logical relations characteristic of living as contrasted with organic systems, and further with a consideration of any implications this might have for general philosophy." So we have the impression here that Waddington is talking about philosophy of biology as philosophy of nature, looking for this big picture, for these general principles governing the living world, the structure, the development, and the behavior and the evolution of living systems. So I think that here is where philosophy of biology and biology meet. That's really a bridge and even overlap between the two. So what I want to say is that, coming back to the cartoon I showed you at the beginning, philosophy of biology and biology are involved in the common task, which is to solve biological enigmas, more experimentally for biology and more conceptually for philosophy. But still the two dimensions are present in both, because philosophy has to confront its result to the empirical and biology to the conceptual. So now, after this pre-introduction, here is the outline of the talk. I'm going to say a few words about the study of biological noise, really basic things you all know better than me. Then I will make some terminological specifications and will raise some philosophical issues about it. And then I will, in two steps, I will concentrate on the relationship between biological stochasticity and explanation. And finally, the fifth part, I will talk you about this philosophical proposal I elaborated with Mark. So first of all, the introduction. So in fact, the expressions developmental noise and biological noise were introduced in the 40s for the first time in biology. I made a quick research before coming here. But then it's in the 50s and 60s that biological noise has been started to be studied, in particular in the context of the study of morphological asymmetries between the two sides of bilateral organisms, like number of bristles in Drosophila or the geometrical patterns of wings of butterflies, and also the study of morphological differences between clonal organisms in a homogeneous and stable environment. And so the hypothesis was that these fluctuating asymmetries, as they were called, and they are still called like this today, and these morphological differences are due not to genetic or to environmental modification, but to some internal perturbation fluctuations affecting the process of development. Then in the 70s and 80s, stochastic mathematical models and computer simulations were started to be used to study development, to model development, and this came to support the hypothesis I just talked about fluctuating asymmetries. And starting from the 90s until now, a consensus rise about this idea that the origin of developmental noise lies in microscopic random events affecting, in particular, gene expression and all the other processes involved in development. And this has been based on both theoretical and experimental research. And so there had been a lot of focus starting from the 90s on noise in the process of gene expression, as you know. What about the definitions of noise in biology? In the biological literature, you can find many verbal definitions of noise, and you can. It's just a sort of synthesis I made about the way noise is defined, like all the time talking about fluctuations or perturbations that are non-genetic and non-environmental, that take place inside. At the intracellular level, at least inside the organism at other levels, and that can have an impact on the process of gene expression, on cell differentiation, and more broadly on the developmental process. So you always have this way of characterizing noise. But then there's a more formal definition, you all know, and it's the way, in fact, noise is often defined using the way it is measured. What do you measure? Mainly you measure the random variation around the mean value of, for instance, gene expression. So around the average amount of some molecular product, like transcripts or proteins produced inside the cell, in the absence of any genetic and environmental change. And so this statistical measure, which is the coefficient of variation, the standard deviation divided by the mean, has been has become the way noise is defined. In fact, noise is often defined in this operational way in terms of, in fact, its results, this random variation around the mean in terms of the amount of molecular products produced. What about the causes? The causes are, as you know, the fact that the intracellular milieu is a Brownian world. This statistical effect called the low number effect, the fact that the few molecules are involved in gene expression, so the fluctuations can have a higher impact. The fact that the intracellular environment is not homogeneous, and even quantum fluctuation can be at the origin of biological noise. And what about the consequences? But the consequences are, in fact, what the definition, the operational definition is talking about, this random variation, which can be measured at different levels. You can measure it at the level of the number of transcripts and proteins produced, of course, in an individual cell over time, or in a population of clonal cells at some point in time, or it can be at higher levels of the phenotype. You have two historical examples. One is the On the left is the figure is representing the stochastic distribution of cell states in Bacillus subtilis, and the fluorescence reveals subpopulation of bacteria that are competent, in which there's a threshold of the expression of the COMK gene, and so a threshold of in the abundance of the COMK protein. And on the right, you have also examples of differences between the fingerprints of twins, or differences in the coats between cloned cats. So these are nothing new for you. I mean, you know all that. Now some terminological specifications and some questions also I have for you. So you have many ways in the literature that noise is qualified in the literature. So you can find expressions like molecular noise, biochemical noise, intracellular, expression noise, splicing noise, developmental, phenotypic, etc. My impression, but we can discuss about that, is that in fact we are talking maybe about the same phenomenon, which is observed and measured at different levels. It's just at which level you look at this phenomenon that leads you to qualify it in this different way: molecular, biochemical, intracellular. We are still talking about the same phenomenon, but we can discuss about that later. And the other terminological point I would like to make is that starting from now, I'm going to stop talking about noise, and I will just use the term stochasticity. Why? It's pretty simple, because noise is a term which has a priori negative connotation. It refers to perturbation, to error versus a signal, relevant information. And second, because it's a term which is entrenched into a Newtonian, as Ignazio would say, or deterministic biology. And so I would like to be non-committal to all that and just use stochasticity as a more neutral term. So philosophical issues about biological stochasticity. I'm going to spot two sorts of philosophical issues. First of all, there are ontological issues like this one. We can ask, what is stochasticity? And this is a question about its nature, ontologically speaking. The ontology is the study of what exists in the world, the furniture of the world. And so we can ask, what is stochasticity? Is something that really, it's something real existing in the world? And so when we talk about stochasticity, we refer to some objective chance, something that a property of the world, of biological processes. And so the probabilities we use to model these stochastic processes has to be interpreted objectively, saying something about the properties of these biological processes, or stochasticity is just a name for our ignorance, with the fact that we don't know and we cannot know all the causes intervening at the origin of all these processes, biological processes we study. And so we refer to some epistemic chance, and the probabilities are not telling us something about the phenomenon, the real phenomena, but something about our degree of knowledge about them. This can be an alternative. For some philosophers, not only about biological stochasticity, but in general about chance, it's an alternative. And it's related to also the question of the deterministic or indeterministic nature of biological processes. Some philosophers would say, if you think that there's objective chance, that stochasticity, biological stochasticity is something real, existing in the world, then it follows that biological processes are indeterministic. And on the other way around, in the case of stochasticity as epistemic chance. But I think that there's a third way. It's too simple, this alternative. There's a third way. I'm not the only one to think in this way. We can also maintain that as far as modelers systematically choose to adopt a certain level of description and explanation, when you do models, you simplify, you abstract, you make abstractions. And so you choose a certain level of description and explanation to account for a phenomenon according to your epistemic interests and goal. So what do? You choose to take into account certain causes, and you choose to ignore other causes. It's not that you ignore them. Some of them you ignore them, but many you just choose to ignore them because they are not relevant with respect to your epistemic interest and goal. And so if you start from this idea, we can say that our best stochastic models tell us something real about the systems undergoing study at the level of the analysis chosen by you because of all these considerations. So we could say that stochasticity is objective as it tells us something real about biological processes. Of course, you ignore something, but then you made choices and you chose to ignore some causes. So this is, I think, an interesting third way to explore. But then you can ask, you are talking about stochasticity, what it is stochasticity. And so first of all, I want to, so on the right, I want to distinguish what we can call a deterministic from a stochastic scenario. In a deterministic scenario, you start from, you have an instruction A producing some outcome X, and then the instruction changes, and instruction B, a specific instruction giving rise or producing some specific outcome. So this is the deterministic scenario. In the stochastic one, the difference is that you have, at the beginning, the initial condition is the same instruction, A, and it can give, or it can give rise to different outcomes. So more than one possible outcome starting from the same instruction. So when I talk about stochasticity, really, I mean, simply I'm not going to give you some mathematical definition. There are many. I mean that it's a situation where from a given situation at given time, different outcomes can take place at a later point in time. So for instance, from the same set of genes in a given environment just before the initiation of gene expression, then you can have quantitative differences in gene expression or of the same gene, or the expression of different genes leading to differences in cell fate. So the idea is that from the same subset of initial conditions, more than one possible evolution. There's more than one possible evolution. And the term subset is very important because of what I said before. I don't want to commit to determinism, undeterminism, all these ontological issues. I think as a philosopher that when you do philosophy of science, you first have to study what you do, science, and then maybe you can infer some ontological thesis. We cannot consider the whole set of initial conditions. We don't do that. So let's be no committal to any philosophical ontological question and just say from the same subset of initial conditions that have been chosen for epistemic reasons, more than one possible evolution is there. So stochasticity is when something can be otherwise, starting from the same subset of initial conditions. So this is for the ontological philosophical issues, and then there's another sort of philosophical issues which are epistemological, more related to our knowledge about it. And one of these issues is the following: what's the relation between stochasticity and explanation? And so, as many other phenomena, stochasticity can be conceived of in two ways. As part of what in philosophy we call the explanandum. What is an explanandum? Is a sentence describing a phenomenon to be explained. So we can consider stochasticity as something we want to explain, in short. Or stochasticity can be conceived of as part of an explanans, a sentence describing a phenomenon that explains. So in short, we study stochasticity and we want to see whether it explains something else. And I'm going to talk about these two conceptions of stochasticity in the two following parts. So first, biological stochasticity as explanandum. So something that we want to, that is to be explained. So when we take this perspective, we ask, what is it, stochasticity? Where does it come from? This is, in fact, in a way, an ontological question. And it is an issue which is addressed in the biological literature. There's this alternative between what biologists call the error hypothesis and the adaptive hypothesis about the origin of biological stochasticity. The error hypothesis that stochasticity is just a matter of a question of molecular errors in gene expression, and thus it is non-adaptive. And in this review, Zhang and Xu, they define molecular errors as any fitness-reducing molecular event. Maybe you don't all agree with this definition. And according to the adaptive hypothesis, stochasticity is adaptive. It conveys a fitness advantage, and I quote Zhang and Xu, it's necessary to support complex forms. So first, I have a question. I have no real answer to that, but are these two hypotheses really exclusive as they are presented in many papers? Maybe not all of them. I haven't read all of them, because we could think that both hypotheses can be valuable and relevant in different circumstances, depending on the specific biological phenomenon concerned. In some cases, biological stochasticity is an error, just an error, and in others, it turns out to be adaptive, or it's really a phenomenon that has been selected in order to produce variability, for instance. It's not one or the other, I think. So we can discuss about that. But then I want to focus on the adaptive hypothesis, which is, I think, the most interesting, I mean, the most challenging. And so the idea is that biological stochasticity in gene expression, for instance, is a trait that is likely to evolve by natural selection. So here we have a selective evolutionary explanation. Then it means that the three conditions, as Darwin, first of all, and then, for instance, Richard Lewontin in the paper in the 70s, the three conditions for evolution by natural selection should apply to biological stochasticity, which means that you must have variation in biological stochasticity amongst individuals of a population. This variation have to cause fitness differences, and also biological stochasticity have to be heritable, inheritable trait. So these three conditions have to apply in order to have evolution by natural selection, in order for biological stochasticity to be a trait likely to evolve by natural selection. One example is an old one. Sorry for the old examples, but I think, I mean, I read more recent papers, and I mean, the way, I mean, the kind of study, there are more and more specific studies, as Marco made me notice. But I mean, this study is perfect to talk also about what is done today. So you have this famous example about the bet hedging competence in Bacillus subtilis. I briefly talked about that before. I think you all know that, but you know, you have the phenotypic state of each individual bacterium depends on the level of expression of this COMK gene that randomly fluctuates over time. So this gene is involved in the production of COMK protein, whose abundance in each cell, as a consequence, randomly varies. And so you have a competent and non-competent bacteria, depending on the fluctuating abundance of COMK, which is a random fluctuation. According to, for instance, Mahamar et al., they are restating this adaptive hypothesis. They say, I quote, our results imply that noise properties are subject to evolutionary forces and suggest how cells might alter those rates to increase fitness. So all this literature about the adaptive hypothesis of biological stochasticity makes me think of what was done many years ago about genetic mutations and all these mutator mechanisms rising the mutation rate in stressful conditions. And I think it's really analogous to what today, well, since the 90s, is argued about noise as an adaptive way to produce variability, and so to enhance the chances for a population of, for instance, bacteria in this case, to adapt to some stressful condition. So I think it's something maybe to look more at previous literature about mutator mechanism can be interesting and inspiring to solve some problems, some open issues about biological stochasticity, even though we are not talking about the same kind of process. But then the question I asked myself is the following: is this adaptive hypothesis really legitimate and well-grounded? I think it's really difficult to have a strong argument in favor of it, because the answer to this question I ask, it depends on several thorny issues. I think some of them are still open. You will tell me. First of all, there's the question of the level of selection. At which level does the selective process at the origin of stochasticity take place? And this is the question of the units of selection, which can be, for instance, a gene, an individual organism, or a group. Then related to this first question, there's a question concerning the identification of the entity that is the beneficiary of the adaptation. Because in fact, the beneficiary of the adaptation does not necessarily correspond to the biological unit of selection. For instance, you can have selection for a certain level of biological stochasticity, and this can take place at the level of individual organisms. Some of them survive and reproduce more than others because of the selective advantage provided by stochasticity in gene expression, for instance. Nevertheless, at the same time, the real beneficiary of the adaptation can be the group, because the presence of variation among individual organisms composing it, so this variability that can enhance the chances to find the right way to solve the challenge, the environmental challenge. Third, there's the question of, I raised before in the previous slide, is stochasticity an heritable trait that can be transmitted from one generation to the next? And if so, how? Is it genetically? Is it epigenetically? It is both. I think there are people working on that. Fourth, more specifically, if you consider developmental stochasticity, for instance, as a result of selection at the level of groups and not the level of individuals, but that's the question of whether the selective process at the level of groups can really take place and be effective. And this is, of course, an empirical question, not a philosophical one. And for instance, you have to check that the advantage that the trait provides to the group is higher than any conflicting selective pressure at the individual level. And also, group selection is still, I think, a controversial notion in biology, even though in the 60s and 70s there was a sort of rejection of it by Williams in particular, but it's a thorny issue still. And last but not least, think about our workshop title, Origin, Maintenance, and Evolution. I would have had Origin, Diffusion, Maintenance, and Evolution. The fact that you can have different, these are different phenomena. That's the origin of biological stochasticity, that it's diffusion in a population, it's maintenance, if it's a trait. And so you can, for instance, say, okay, the origin is just a physicochemical origin, but then its diffusion is due, for instance, I don't know, in a small population to drift, genetic drift, and then its maintenance can be thanks to evolution by natural selection. So you can have different kinds of explanations to account for different aspects of the evolution of biological stochasticity. So even if it's tempting, it's just what I want to say here, and promising to maintain the adaptive hypothesis about biological stochasticity, it's not so straightforward to corroborate, as you know, of course. Now let's talk about biological stochasticity as explanatory, as something that is not to be explained, but something that explains something else. And so here the question is, what is its explanatory role in biological models? By the way, you have to know that in philosophy there's debates about, because we talk about stochasticity, we talk about chance, and there's a debate still open about whether chance can be explanatory. Some philosophers, like Paul Humphreys, who thought that explanation is causal, also thought that chance cannot be explanatory because it's not causal. Other people suggested, on the contrary, we can define chance in causal terms, and so it can be explanatory. Other people argue for an explanatory role of chance, like Marco, not in causal terms, but in other terms. So there's still an open debate. So the question of whether stochasticity is explanatory is a debated question in general in philosophy of science, and I think in particular in the case of biology, because of all this research about biological stochasticity now. So you can answer to this question, which is an epistemological question, saying that stochasticity just explains our ignorance. It's a sort of epistemic chance. Or it's not explanatory, but it allows us to make better predictions. Or you can argue that, in fact, it explains something. And I'm going to focus on this point. And in particular, if you focus on the explanation, you have to show, yes, it explains. But I think that in the literature, you find so many papers showing that stochasticity, biological stochasticity as a role, that it's a good working hypothesis to start. It's a good bet to say, yes, it can explain something. And then I'm going to focus on the question of what does it explain? And Marco, I think, is going to talk about how stochasticity can explain something in biology. So, first of all, we can say just very simply that stochasticity, biological stochasticity, allows us to see and account for new variation, new in the sense of variation that we ignored before. Think about Jacques Monod. Jacques Monod he said that he conceived a population of bacteria as an ideal gas, just all the molecules the same. This is not the way we look at a population of bacteria today. So another proposal is to say that biological stochasticity participates to the explanation of individual development, and in particular, it accounts for the uniqueness of each individual organism. It accounts for why each individual organism is as it is, with specific species-specific traits, and in particular, unique traits. And the same you can say for individual populations. But here you can ask me, it's not important to account for unique traits. I mean, should it be the task of developmental biology to account for the uniqueness of each individual organism? It's not what we do. At least traditionally, this is not the aim of developmental biology, which classically focuses on the set of mechanisms common to all individuals of the same species and that are at the origin of common species-specific trait. So here is a quote by Richard Lewontin saying that, yeah, developmental biology is not interested in this uniqueness. Classically, traditionally, is arguing for the other way around, but yeah. Classically, developmental biology study development as a type of process, as a general sort of process. And so we can ask, what I'm suggesting is development as a token, as a particular concrete instance, a relevant explanatory target of developmental biology. And if so, I mean, I think it is, it should be, but if so, developmental biology could and should be conceived both as a nomothetic science, a science which aims is to make generalizations about development as a kind of process, and as a historical science, about studying the construction of a single individual organism, a token, where the history and thus stochasticity matters, is relevant. And if you don't think that developmental biology should study development as a token, I can still tell you that, and I mean, there are many other papers. I took this one by Adrian Roeder. I like this paper a lot. The stochasticity can also contribute to the explanation of development as a type, and in particular, to the explanation of one of its properties as a type of process, its robustness. Roeder says, I quote, developmental robustness counterintuitively actually requires variability as well as stochasticity at its origins. Sorry, that's an error. Why does she says counterintuitively? Because if you think about stochasticity in terms as noise, then it's counterintuitively to say that noise explains robustness, because noise is an error, it's some perturbation. How can it explain robustness? But then this is what she shows and she argues for. She argues that in order to cope with unpredictable environmental perturbations, so developmental robustness requires variability and stochasticity. And her example concerns cell differentiation and stress, so in plants. In stressful environmental situations, stochasticity can be a source of variability, as you know, in particular at the level of gene expression, and during cell differentiation, and more generally during the cell cycle, the external and internal environment can change abruptly. And by generating variability in terms of, for instance, proteins produced, stochasticity in gene expression can thus help the cells to find new survival strategies. So stochasticity sometimes itself is used as a mechanism to promote robustness. So in this sense, you can argue that stochasticity can also explain some property of development as a type. Its robustness, its stability, which can be seen as something in opposition to stochasticity. And now I'm going to conclude the talk with this proposal we have started to elaborate with Marco. It's really, an abstract one, trying to, to find out whether it's useful or not. So we are going to see. So first of all, as you know, there's more and more evidence for the role of stochasticity in gene expression at different levels in different organisms, from virus to, not organisms, but from virus to other organisms like prokaryotes, eukaryotes, etc. Here you have a list from historical papers at the beginning of 2000. So there are many, many different phenomena where this role of stochasticity has been shown. And you can add, I could have pages about that with references and phenomena where stochasticity is involved. And then I want to make a short reminder here. Think about what I said at the beginning, the fact that often biological stochasticity is defined in operational terms, in terms of results, and that we know its physical causes. But then we should ask what lies between the two, the causes and the results. Because we know the causes and we measure stochasticity and define it in terms of result, but what lies between the two? We should think of stochasticity as a process over time, taking place over time during development and contributing to its progress over time. And that's why I think in order to improve our study of stochasticity, maybe we should go back to Waddington again and to his idea of development as epigenesis. So epigenesis versus the idea of preformationism is the idea that development is a process that takes place over time, and the organism is not preformed, even in terms of genetic information, it's not preformed, but it's progressively constructed throughout time due to various kinds of developmental interactions. And so the question Waddington asked to himself was how the genotype can bring about phenotypic effect. And his answer was what we call today epigenetics. So the study of all these processes which dynamically organizes and constructs tissues, organs, and entire organisms by interacting with the genotype and reacting to the external environment. And according to him, stochasticity was the component part of what he called the epigenotype, this set of dynamic structure system of processes. So stochasticity, according to him, was a component part of the epigenotype and of the epigenetic landscape. So here are two famous representations of the epigenetic landscape. So the first one was a representation made by an artist, a friend of Waddington, John Pipel. And then you have the famous one on the right. So I'm going to be brief, but it's a system, it's representing a system of valleys diverging down an inclined plane. The inclined plane symbolizes the tendency of developing piece of tissue to move forwards a more adult state, or you can think about the ball as an undifferentiated cell, for instance, going down and differentiating. And then underneath you have pegs, and the pegs represent the genes, and then you have the gyropes representing the interaction between genes and between genes and environment, and their chemical, what you know, cause they chemical activities in a way. And what is underneath all these interactions shapes, give the shape to the developmental landscape, the shape we can see, the system of value we can see above. And so the ball, the cell going down takes different value. It can change the value because of perturbation, for instance, and differentiate going through a path or through another one. And so, as I've just said, Waddington included the stochasticity in the epigenetic landscape. He called it developmental noise. And he characterized it as slight irregularities in interaction between different parts of the germ, as something that cannot be attributed either to genetic factors or to environmental circumstances, as some looseness or play. Also, he said about developmental noise that is something that varies somewhat around the mean value. Noise as the lower limit of the variation. So, and he really in the representation, he puts stochasticity, noise, developmental noise, in the representation of the ball. He says, I quote, if canalization is represented as a valley in an epigenetic landscape, the noisiness of the system might perhaps be symbolized by the imperfection of the sphericalness of the ball which runs down the valley. So, anyways, we can conclude that today we are not saying something new about noise. It's not genetic, non-environmental source of variation. It's internal and unpredictable. It's some deviation with respect to some average. It's inherent and unavoidable. It's characterized in negative. So my proposal, I think that we should use the epigenetic landscape by Waddington and try to look for a new place for stochasticity in Waddington's epigenetic landscape in the light of what we know today. Waddington was many, many decades ago. So use Waddington's epigenetic landscape because it is a metaphor that has no particular grounding in physical reality. It's a visual image, but this image had, and it continues to have, a big heuristic value. For instance, a colleague, Jan Betke, philosopher of biology, showed in a very nice paper I suggest you to read, that Waddington's epigenetic landscape has been a tool for visualization, a tool for transdisciplinary communication, a creativity tool, a modeling tool, a way to coordinate, in particular, methodological strategies and modeling approaches. So we have to exploit this heuristic device in order to, I think it's our proposal, in order to better think about and make sense of biological stochasticity. And so our proposal, Marco and I, is to use this heuristic tool to better think and understand the role of stochasticity in development, to relocate stochasticity in the epigenetic landscape in accordance to recent results. So the question we raise is, how could stochasticity be thought within the landscape framework? Where should stochasticity be located in this landscape? In what spatial relation does stochasticity stand with respect to the other components of the epigenetic landscape? And we have a suggestion. It's really a sketchy one. Maybe it's the wrong one, but it's just a way to start discussing about that. So we suggest that stochasticity as a set of processes. So we have to think about it, as I said some slide before, as a process, what lies between the causes and the results, takes place underneath the epigenetic landscape during development, in the underneath part. It affects the length, the tension, and the bend of ropes, that are the chemical activity tendencies of genes, and it changes over time in development and evolution. So here it's a draw made by Marco. So you have to think that according to Waddington, the epigenetic landscape is there. During a development of an individual organism, the epigenetic landscape is already there, and the ball goes down through a valley or another. The idea is that the epigenetic landscape, first of all, is dynamical. Is dynamically built up over time during development. And stochasticity, you cannot see it very well here, is symbolized in the epigenetic landscape by a sort of web underneath, a sort of spider web, but a very disordered spider that is continually waved over time. And so its dynamical construction affects the length and the tension and the bend of ropes, and so the shape of the landscape. So here you can see it more. It's my draw underneath. So it's not really, it's just a sketch. It's not really perfect at all. So stochasticity is represented by this web that builds up over time of the course of development. And initially this construction has a greater impact on the activity and interaction among genes. But as the web takes shape and its form stabilizes, its construction has less impact on the guide ropes. Like what has been shown, for instance, in plant development or in the Drosophila development, the fact that stochasticity has a higher impact at the initial steps of it, and then less impact later. But then, as I discussed with Luisa yesterday, there's this hourglass model. So we should also think about that, the fact that stochasticity can also follow this developmental hourglass model. And so we can also ask how to represent the developmental hourglass model in Waddington's epigenetic landscape. So what's new if we think about stochasticity and replace it in this way in the epigenetic landscape? The epigenetic landscape is no more static, but ontogenetically dynamic. And stochasticity is conceived of as an epigenetic process with other epigenetic processes like DNA methylation, histone marks, chromatin structure changes, etc. Stochasticity is more than just noise. It's a component of the developmental process. And so we should have we have to model it as a variable describing the behavior of biological system, not just a narrow term. And then there are still open questions, many. More questions about the properties of stochasticity. Are there different kinds of stochasticity? How the properties of stochasticity change over time? What are the relationship between the genetic and epigenetic components and stochasticity? How and to what extent can stochasticity change the epigenetic landscape, etc.? So many questions. You have even more, I think. And the epistemic gain, I'm going to conclude that it's my last slide, of doing that is really to, I think it can be promising, but maybe I'm wrong, to update this visual metaphor that have been really useful and fruitful over the years since its introduction by Waddington, even outside biology, even in physics, for instance. So this visual metaphor whose legacy and heuristic potential has been really broad in life sciences and beyond. And in line with Waddington's visual metaphor strategy, to have an intuitive understanding of biological systems, to make it a contact within the unknown, stochasticity in a rather wide way, but which can be useful then to develop more precise and formal descriptions and models of stochasticity in biological systems. So that's all. Thank you very much for your attention. I'm really glad to.